Some presynaptic terminals in the central nervous system appear non-functional or silent. Here we present a protocol for identifying silent presynaptic terminals. This protocol can be applied to identifying treatments and manipulations that are suspected of silencing or awakening terminals. The protocol combines vital dye labeling of synaptic vesicles during neuronal depolarization with subsequent labeling of fixed cells with an antibody that labels all presynaptic terminals, both silent and active. Synapses co-labeled with dye and antibody are active. Synapses labeled only by antibody are silent.
Hi, I'm Amanda Taylor from the laboratory of Dr. Steve Minerick in the Department of Psychiatry at Washington University School of Medicine.
I'm Xiaoping Jiang, also from the Minerick lab.
Today we would like to show you a procedure for visualizing silent presynaptic terminals using cultured hippocampal neurons.
We use this procedure in our laboratory to study how electrical activity and other signaling pathways can modulate the neurotransmitter release.
So let's get started.
In a sterile laminar flow hood, use hippocampi from postnatal day 0 to 3 rats or mice to prepare dissociated cell cultures according to our published protocol. Our cultures are mixed neuronal glial cultures derived from the same dissection. Cells should be plated in sterile 35 mm dishes, each containing a number 0 cover slip that was coated with 5 mg per milliliter of collagen. Plate the neurons at an approximate density of 500 cells per centimeter squared. 12 rat pups will typically yield 30 35 mm dishes. Allow the cultures to grow in a culture incubator for 10 to 14 days for synaptic development and maturation. Add the antimitotic Ara-C to arrest glial growth at day in vitro 4. Also, at day in vitro 5, perform a half-medium exchange with neurobasal medium plus B27 supplement to enhance neuronal survival. During this maturation period, introduce treatments that alter the number of presynaptically silent synapses. We find that one convenient way to silence a large percentage of glutamate synapses is a 4-hour treatment with 30 millimolar potassium. When the cultures are mature, neurons should have a relatively low density with well-separated and extensive neuritic processes. Remove the cultures from the incubator and bring them to the bench. Replace the culture medium with a buffered saline solution containing 45 millimolar potassium chloride and 10 micromolar FM143FX dye. This solution should also contain NBQX and APV to prevent recurrent signaling. It is important that the FM143FX stock solution and experiments be kept in the dark to prevent photobleaching, so cover the culture dishes with foil for all subsequent incubation steps. Incubate for exactly 2 minutes at room temperature. Remove the dye solution and wash for only 3 to 5 seconds in heaps buffered saline with 500 micromolar Advocep 7 to remove nonspecific dye. Timing in this step is critical, as prolonged exposure to Advocep 7 will remove all the FM43FX labeling. Then wash the cells in heaps buffered saline without Advocep 7 for 5 2-minute intervals. In a chemical fume hood, fix the cultures in 4% paraformaldehyde and 0.2% glutaraldehyde in PBS for 10 minutes. Wash the cells briefly with PBS. The cells can now be moved out of the fume hood. Add blocking solution containing 4% normal goat serum and 0.04% Triton X to reduce background and permeabilize the cells for immunostaining. Incubate for 15 minutes. Stain the cells with vGlu-T1 antibody diluted to 1 to 2000 in blocking solution. Incubate the cultures in antibody with gentle rocking for 3 hours. Wash the cells 4 times with PBS. Incubate the cells with Alexa 647 conjugated anti-guinea pig antibody diluted to 1 to 500 in blocking solution. Maintain the cultures in the secondary antibody with gentle rocking for 30 minutes. Wash the cells 4 more times with PBS. Place a small drop of Floramount on a clean glass slide. Using forceps, pick up a cover slip and gently lower it onto the drop of Floramount, cells facing the slide. Allow the slide to dry overnight. Prepare a 60x oil objective on a confocal laser scanning microscope. Place a slide on the stage so that the cover slip is facing the objective. Find a field of neurites that is not overly dense and that is away from the soma, which can retain residual dye. Zoom to a field size of 51 by 51 microns. Using image acquisition software, acquire a Z-stack covering a depth of 7.8 microns over 27 steps of 0.3 microns. For each step, scan first for the vGlu-T1 antibody to identify all glutamatergic synapses in the field, then rescan for FM143FX fluorescence. To analyze images after acquisition, use analysis software to convert the Z-stack into a composite single-plane image based on the maximum intensity reading from any plane at each pixel. Set threshold to reduce background. Identify vGlu-T1 terminals without reference to the FM143FX stain, and mark these puncta as regions of interest. We typically select 10 puncta per field. Repeat thresholding and puncta selection on the FM143FX image, and superimpose the images to identify silent synapses. An absolute pixel criterion, or a percentage pixel criterion, can be used to distinguish active, FM143 positive, from inactive, FM143 negative synapses. In our cultures, we find that 70 to 80% of glutamate synapses, defined by vGlu-T1 staining, exhibit detectable FM143 staining. In overlaid images of green FM143 fluorescence and red vGlu-T1 staining, this is evident as an abundance of yellow puncta, synapses with co-localized markers. These are indicated by the arrows. Approximately 20 to 30% of red vGlu-T1 positive puncta will fail to have green FM143 fluorescence above baseline. These are the inactive presynaptic terminals, and an example is indicated by the arrowhead. A third population of synapses will also be observed. These are positive for green FM143 fluorescence, but devoid of red V-gluT1 staining. These are active GABAergic synapses, and an example is indicated by the asterisk. These are excluded from most of our analyses, but can be explicitly studied using a vesicular GABA transporter antibody in place of the V-gluT1 antibody.
We have just shown you how to visualize presynaptically silent synapses using FM143 and V-glut1 immunostaining.
When performing this procedure, it is important to remember that FM143FX is sensitive to both the time in the adverse F7 wash and the concentration of the treating X100 included in the blocking solution.
So that's it. Thanks for watching and good luck with your experiments.